PCR, or polymerase chain reaction, is not just about amplifying DNA. It plays a crucial role in research, medicine, forensics, and even agriculture. Gene expression varies across different cell types, tissues, and organisms. Neurons and muscle cells, though they share the same DNA, express different genes at different levels. But how do scientists measure these differences? That's where RT-PCR comes in. To analyze gene expression, we first extract RNA from a sample. Since RNA is unstable, we convert it into complementary DNA, or cDNA, using an enzyme called reverse transcriptase. This process is known as reverse transcription. Once we have cDNA, we amplify it using PCR. The sample goes into a thermocycler, where it undergoes cycles of heating and cooling. This allows DNA to denature, primers to bind, and new DNA strands to form, doubling the amount of cDNA in each cycle. But how do we measure gene expression? In real-time PCR, also called qPCR, fluorescent dyes bind to the amplified DNA. The intensity of the fluorescence corresponds to the amount of gene expression in the sample. By comparing samples, we can determine whether a gene is upregulated or downregulated. So what does this mean? Increased fluorescence in neurons indicates higher gene expression. In simple terms, the gene is actively producing more messenger RNA in neurons compared to muscle cell. This suggests that the gene plays a more significant role in neuronal function. Why is RT-PCR chosen over northern blotting and other techniques? RT-PCR is the most sensitive method for detecting and quantifying mRNA. Unlike northern blot analysis and RNase protection assays, RT-PCR requires much smaller sample sizes and can even quantify RNA from a single cell, making it the preferred choice for precise gene expression studies. In research, RT-PCR is a powerful tool for measuring gene expression. Take the study by Lin et al., where scientists investigated the gal genes in yeast. They introduced a mutation in a regulatory protein, suspecting it would abolish gal gene expression. To confirm this, they used RT-PCR to compare gene expression in normal and mutant yeast cells. The results? The mutation drastically reduced gal gene expression, proving its role in regulation. To further validate their findings, they performed northern blot analysis for detailed RNA profiling. This study highlights how RT-PCR helps uncover gene functions at a molecular level. How does RT-PCR help insert eukaryotic genes into prokaryotes? Eukaryotic genes contain introns, segments that must be removed before producing a functional protein. But here's the problem. Prokaryotes like E. coli lack the splicing machinery needed to remove introns. This is where RT-PCR comes in. Scientists first extract mRNA from eukaryotic cells. Since mRNA already has its introns removed, we can use reverse transcriptase to convert it into complementary DNA, or cDNA containing only exons. Next, this cDNA is inserted into a bacterial plasmid and transferred into E. coli. Unlike genomic DNA, this version is ready for direct protein synthesis, so no splicing is required. Once inside the bacteria, the gene is transcribed and translated efficiently, producing the desired protein. RT-PCR is a powerful and precise tool for studying gene expression, offering advantages over traditional methods like northern blotting. From research applications to gene insertion, its impact is significant. Don't forget to like, share, and subscribe for more. Until next time.